Between 2014 and 2015, the world basically held its breath as the West African Ebola epidemic unfolded. I mean, we're talking about an outbreak that caused over 10 times as many cases as all previous Ebola outbreaks put together. It was a massive wake-up call, right? We saw firsthand how easily one of the deadliest pathogens on Earth could just leap across borders, rivers, and even oceans. But how much do we actually understand about this virus? Like, how does it attack our cells? And most importantly, how exactly is modern science fighting back?
Welcome to today's Explainer. I'm absolutely thrilled you're tuning in. Today, we're going on a seriously fascinating journey to demystify Ebola. We're going to take its terrifying reputation and look at the real story, a story of incredible medical mastery. We'll dive deep into exactly how this virus operates on a microscopic level and the game-changing treatments we now have to stop it in its tracks.
Here's our roadmap for today. First up, we'll meet the Ebola virus. Then we'll uncover where it hides and how it spreads. Chapter 3 takes us inside the human body, followed by Chapter 4 on how we fight back. And finally, we'll look at the road ahead. Let's jump right into Section 1, Meet the Ebola virus, History and Taxonomy.
So the story officially kicks off in 1976. That's when the very first simultaneous outbreaks happened in Sudan and Zaire, which we now know as the Democratic Republic of the Congo. The virus actually takes its name from the Ebola River, which is right near that original village in Zaire. Now, the way we classify the virus has evolved over time. It was introduced as the Ebola-like viruses genus in 1998. Fast forward to 2014, we hit that massive, historic outbreak in West Africa. And then just recently in 2023, the virological genus was officially renamed to ortho-ebolavirus. Kind of a mouthful, but that's the official name now.
Now, here's something a lot of people don't realize. Ebola isn't just one single virus. It's basically a family. There are actually 6 distinct species within this genus. The Zaire species is definitely the most notorious. It has the highest mortality rate and is the culprit behind most of the major outbreaks. But there's also Sudan, Bundibugyo, Reston, which is interesting because it affects other primates, not humans, and Thai forest. You'll also spot the newest addition of the family down at the bottom, Bombali Ebola virus, which researchers recently isolated from bats in Sierra Leone. Moving on to Section 2, Where it hides and spreads, the reservoir and transmission. Okay, so this brings us to a really crucial concept, the spillover event. Basically, this is when an infected animal directly passes a virus to a human. Scientists strongly believe that African fruit bats are the primary natural reservoir here. That means the bats maintain the virus in what's called an enzootic cycle. It just constantly circulates within the bat population, hanging out there until that fateful moment of direct contact with a human or another animal. So once it actually reaches humans, how does it spread? I want to ask you this directly because it's probably one of the most common myths out there. Is Ebola airborne? The answer is a definitive no. There is a huge difference here. Respiratory viruses like COVID-19 or the flu, yeah, those can spread just by breathing the same air as an infected person. Ebola does not work like that. You absolutely cannot get it just by being in the same room as someone. It strictly requires transmission through infected bodily fluids, period. Let's break down exactly what that means. Human-to-human transmission happens through direct contact with broken skin or mucous membranes. We're talking about blood, sweat, vomit, feces, fluids from the sick or deceased. It can also spread through contaminated objects, like needles or even unwashed bedding. Direct contact during traditional burial ceremonies is actually a massive transmission route, as is handling or eating infected wildlife, often called bushmeat. It can even be transmitted via semen from a recovered patient. But the golden rule to remember here is this: a person remains infectious for as long as their blood contains the virus. Let's dive into Section 3, Inside the Human Body, Pathogenesis and Symptoms. All right, so what happens on a cellular level? It's essentially a microscopic heist. First, the virus attaches to your host cells using its viral glycoprotein, or GP. Next, it literally tricks the cell into swallowing it up through a process called macropinocytosis. Basically, the cell just drinks it in. Third, once it's inside a cellular compartment called the late endosome, host enzymes called cathepsins start chopping up that viral glycoprotein. And finally, this newly processed protein binds to a specific host protein called NPC1. Think of NPC1 as the master key. Once it binds, the viral envelope fuses with the cell membrane, drops its viral RNA into the cell, and completely hijacks the cellular machinery. Now, after this silent cellular invasion kicks off, there's a waiting period. The incubation period, which is the time from that initial infection to the very first symptom, can vary wildly. We're talking anywhere from 2 to 21 days. But when the disease finally does manifest, it escalates incredibly fast. It usually starts out looking a lot like the flu: sudden fever, overwhelming fatigue, muscle pain, maybe a headache. But then it rapidly progresses to severe vomiting, diarrhea, and intense abdominal pain. From there, patients might develop rashes, impaired kidney and liver function, and in the most severe cases, generalized organ failure. It's incredibly aggressive. Because of this, surviving that acute phase is absolutely crucial. But here is a truly wild fact about the recovery process. Even after a patient survives and the virus is totally cleared from their blood, viral RNA can actually stick around. It hides out in what we call immune-privileged areas. These are parts of the body that are somewhat shielded from the immune system, like the ocular fluid inside your eyes or in semen, and it can hide there for well over a year. This leads us right into Section 4, how we fight back: treatments and vaccines. Despite how scary all of this sounds, humanity is far from defenseless. Back in late 2020, we had a massive breakthrough with 2 FDA-approved treatments for the Zaire Ebola virus, Inmazeb and Ebanga. They are both monoclonal antibody therapies. Ebanga uses a single antibody to block the virus's glycoprotein from entering the host cell, but Inmazeb actually uses a combination of 3 different antibodies. And the way those 3 specific antibodies in Inmazeb work together is just brilliant. Not only do they neutralize the virus by physically blocking it from getting into the host cells, but they also trigger something called antibody-dependent effector function. In plain English, the drug acts like a biological flare gun. It recruits other immune cells to rush in and aggressively clear out the infected cells. It's literally calling in the cavalry. But remember earlier we noted that Zaire is just one species. What about the others? Well, researchers are actively running clinical trials right now in the DR Congo. They're testing an experimental monoclonal antibody called MBB-144. This is a pan-Ebola virus therapy, meaning it targets binding sites that are shared across multiple strains, including Sudan and Bundibugyo. They're also testing the well-known antiviral remdesivir. By going after these unchallenged strains, science is pushing closer and closer to a universal defense. And of course, we absolutely have to talk about vaccines. This search started way back in 1976, and today, we have 2 fully approved vaccines for Ebola Zaire. That is a huge win. Meanwhile, the fight continues for the other strains, with a really promising candidate for the Sudan Ebola virus actively making its way through phase 2 clinical trials as we speak. Finally, let's look at Section 5, the road ahead, future challenges. I want you to consider this quote from the CDC archives. "A disease threat anywhere is a disease threat everywhere." During that massive West African epidemic, we saw how highly mobile populations and porous borders just let the virus spread like wildfire. It's a stark reminder of why understanding and containing outbreaks at their origin matters to absolutely everyone, including you watching this right now. Because pharmacology alone isn't a silver bullet. Public health strategy is the real cornerstone of containment. That means isolating patients in designated treatment centers early on. It requires ensuring safe, dignified burial practices. Crucially, it means involving local communities, having open, honest discussions so they are active partners in the response. We also have to rigorously trace and monitor contacts for that full 21-day incubation period, and work tirelessly to reduce the risks of wildlife to human transmission. Which leaves us with one final, pretty profound question to ponder. The exact species of bat that naturally harbors this virus is actually still under investigation. If we can successfully pinpoint that exact animal reservoir, could we manage the delicate balance between human populations and wildlife well enough to prevent Ebola spillovers before they ever even happen? It's a fascinating frontier for science. Thank you so much for learning with me today, and I really hope you keep exploring this incredible topic.